The liver receives 30% of the resting cardiac output and acts as a giant chemical processing plant in the body. These chemical reactions, called metabolism, are central in the regulation of body homeostasis. The liver cells, called hepatocytes, contain thousands of enzymes essential to perform vital metabolic functions. They are the supermodels in the world of cellular metabolism. The liver metabolizes both beneficial and harmful substances. It stores nutrients and other useful substances, as well as detoxifying or breaking down harmful compounds. These can then be excreted from the body in bile via the liver, in urine via the kidney, or by other means. The liver is involved in the metabolism of nutrients. It receives digestive products in the form of glucose, amino acids, fatty acids and glycerol. The metabolism of carbohydrate, fat and protein takes place in the liver, although specific functions are carried out by fat depots and skeletal muscle. Metabolic end products are often stored in the liver and utilised at a later stage if required. How the hepatocytes deal with the nutrients depends on whether each nutrient is in abundance or whether levels are low in the body and they are therefore in demand. The hepatocytes alter their metabolic pathways accordingly. Glucose is a vital energy source for cells, and levels in the bloodstream must remain constant. The liver helps maintain blood glucose levels in response to the pancreatic hormones insulin and glucagon. After a meal, glucose enters the liver and levels of blood glucose rise. This excess glucose is dealt with by glycogenesis, in which the liver converts glucose into glycogen for storage. The glucose that is not stored is used to produce energy by a process called glycolysis. This occurs in every cell in the body. In between meals or during starvation, blood glucose levels fall. The hepatocytes detect this change and restore glucose levels by either glycogenolysis, which converts glycogen back to glucose, or gluconeogenesis, in which non-sugars, such as amino acids, are converted to glucose. The liver is involved in fat metabolism and synthesizes lipoproteins, cholesterol and phospholipids essential for many body functions. Lipids also provide a valuable alternative energy source to glucose, and so the metabolic fate of fats and lipids will depend on the levels of intake in the diet and energy expenditure. If fat is in excess, the liver prepares fat for storage. Lipogenesis is the metabolic process in which fats, composed of fatty acids and glycerol, are converted for storage in subcutaneous tissue and other storage depots. If energy and glucose levels are low, stored fat is converted back into glycerol and fatty acids by a process called lipolysis. This occurs in adipose cells, but the fatty acids and glycerol are transported to the liver for use as an alternative energy supply. Amino acids are transported to the liver during digestion, and most of the body's protein is synthesized here. If protein is in excess, amino acids can be converted into fat and stored in fat depots, or if required, made into glucose for energy by gluconeogenesis, which has already been mentioned. However, before amino acids can be utilized in these ways, the first step is to remove the nitrogen-containing group, NH2. This very important metabolic process is called deamination. In the hepatocytes, NH2, the amino group, quickly changes into ammonia, NH3, which is highly toxic to the body. The liver acts fast to convert ammonia into urea, that then can be excreted in the urine and eliminated from the body. The liver is vital for the detoxification and destruction of endogenous and exogenous substances that are harmful to the body. The liver's own phagocytes, which reside in the lobules known as Kupffer cells, digest and destroy cellular debris and any invading bacteria. Other exogenous substances like drugs and alcohol are detoxified by the liver. Endogenous substances, or those produced by the body, are also dealt with by the liver. Amino acids are deaminated, some hormones are inactivated, and bilirubin, a product of the breakdown of old red blood cells, is also detoxified and rendered harmless by liver metabolism. The liver synthesises bile, which is important for fat digestion and is also a route of excretion from the body. Bile consists of water, bile salts, cholesterol, phospholipids, electrolytes, and bile pigments which give it its typical yellowy-green colour. Bile is stored and concentrated in the gallbladder. The presence of fats in the gut during meals stimulates the gallbladder to empty. Bile enters the duodenum, emulsifying fats into smaller globules, which can then be broken down further by lipase enzymes. Metabolic wastes and drug products may form part of the bile, which can then be excreted from the body through the digestive tract in the faeces. Bilirubin, the toxic end product of hemoglobin breakdown, is excreted from the body in this way. The liver plays an important role as a storage facility. The hepatocytes take up many types of vitamins and minerals from the blood and stores them. These include vitamins A, B12, D, E and K, and minerals such as iron and copper. Glycogen, which is formed from excess glucose, is also stored by the liver, although muscle tissue can also store glycogen too.